Hello, I'm Dr. Adriana and I'm a research associate at the Coral Assist Lab at Newcastle University in the United Kingdom. Today, I would like to talk to you about coral intrapopulation variability of heat tolerance. So the work that I will be presenting here was conducted by the members of our research group, led by Dr. James Guest. All field work was carried out in Palau in the Western Pacific with the support of the Palau International Coral Reef Center. The coral reefs are facing unprecedented declines due to marine heat waves that result in mass coral bleaching and mortality events. Given the extent of projected ocean warming, heat tolerance is the trait most likely to bolster coral reef resilience. Actively assisting corals to adapt to future warming conditions through innovative management actions include assisted evolution. Assisted evolution aims to increase the frequency of specific adaptive traits across a population to improve their future survivorship. But for assisted evolution to work, we need to be able to select the desirable phenotypes. However, the required knowledge about how to best select these phenotypes is still in its infancy. So here, we address the challenge of identifying heat-tolerant corals able to resist marine heat waves and quantify tolerance variability within a single coral population. If sufficient within population variability in tolerance exists, then using colonies from a target population as the source for assisting evolutionary this population has the advantage of reducing the likelihood of maladaptation to environmental variables and may reduce the risk of selecting different genotypes, variants, or subspecies. It also avoids the logistical problems related to translocated colonies from one population to another. So what we did was we designed an experimental temperature profile to mimic a marine heat wave of 6 weeks duration. NOAA's Coral Reef Watch calculates degree heat in weeks based on a 5-kilometer satellite sea surface temperature and the corresponding dataset of the maximum of monthly mean climatology. We use a linear regression to calibrate the NOAA's 5 kilometers mean monthly mean to the in situ temperature data from loggers at our study site, Masterchur Reef, between the period of 2018 and 2021. We then cross-reference the 5-kilometer degree heat in week profile with the in situ degree heat in week profile to ensure that heat stress was accurately reflected using this adjustment. Applying the adjusted degree heat in week to the temperature data from our experimental tanks shows that by the end of the experiment, we have reached 9 degree heat in weeks, which is enough to cause coral bleaching responses. This profile is similar to past heat stress events at the study site that resulted in coral mass mortalities in 1998 and in 2010. So we sampled 102 colonies of Acropora digitifera, and we took 6 fragments of each colony and exposed to temperature stress profiles previously described in the previous slide. Every second day, visual inspection of the fragments was done, and a health status score was assigned to each fragment. These scores were used to estimate a bleaching and mortality index per colony, using the proportion of fragments in each health score category. This continuous variable was used to compare the phenotypic response of all colonies throughout the duration of the experiment by estimating a mean bleaching and mortality index. To classify colonies into categories according to their heat tolerance, we decided to have low heat tolerant colonies, which were those that had all fragments dead at the end of the exposure. And high heat tolerant colonies were all those ones that had all their fragments alive and healthy at the end of the exposure. So according to these categories, we identified 31 colonies having relatively low heat tolerance, indicated here in this graph with red, and 30 colonies with relatively high heat tolerance. We found considerably high levels of intrapopulation variation in heat tolerance, which is evident from the different mean bleaching mortality values in this graph, where each row represents a colony and their values indicate the mean bleaching and mortality index. Analysis of the Symbiodiniaceae community composition revealed that 97% of the colonies used in the experiment had a single Symbiodiniaceae ITS2 type profile dominated by Cladocopinum with the C40 variant, which is not surprising given that colonies are from the same population. This result indicates that heat tolerance in these colonies was unrelated to the algal Symbiodiniaceae community structure. So using the BMI values for each colony, Liam Lachs, who is one of our PhD students, calculated the variation in heat tolerance as the difference in degree heat in weeks tolerated by the more and the less tolerant colonies from our population. So comparing the 30 high heat tolerance with the 31 low heat tolerant colonies and modeling their bleaching and mortality index as a function of the degree heat in weeks using a binomial generalized linear mixed model. Results show that high tolerant colonies were able to withstand an additional of 2.87 degree heat in weeks compared to low tolerant colonies to reach the same mild bleaching response of 0.38 BMI. This analysis was repeated for progressively smaller sample sizes, where at each iteration, the colony closest to the population mean bleaching and mortality index was removed from each heat tolerant category, and then a new generalized linear mixed model was run. This stepwise reduction was repeated until the upper and lower deciles of the population were reached, meaning that we had only 10 colonies in each heat tolerant group, beyond which sample sizes were too low to draw robust conclusions. At this level, the population variation in heat tolerance was of 4.84 degree heat in weeks between the upper and the lower decile groups to reach the same mild bleaching response. So we wanted to understand what these differences mean in terms of the additional time heat tolerant corals may have until the onset of annual bleaching events. We also wanted to test this under different climate change scenarios. So degree heat in weeks at our study reef were projected from 28 global climate models and for 2 global shared socioeconomic pathways, SSSP2 4.5, which is meeting 150% of the pledges of the Paris Agreement, and a worst case scenario, SSP5 8.5, where the world economy is heavily dependent on fossil fuels. So this work was led by Liam with data provided by Ruben van Hooydonk. 3 bleaching mortality thresholds of 4, 8, and 12, indicated here in blue, yellow, and red, were used based on the approximately 4 degree heat in weeks differences that we found in between the high and the low tolerant colonies to derive for projections of bleaching and mortality conditions for our population. So these results show the projected differences in timing of onset of annual bleaching conditions for our high and low corals. Pairwise comparisons were all significant between degree heat in weeks thresholds. Based on these results, we might expect about 10 years of difference between 4 and 8 degree heat in weeks, regardless of the climate scenario. For corals with a bleaching threshold of 8 and 12, however, the additional time they have before bleaching onset can almost be doubled from 10 to 17 years if there is a strong action on climate change. So in conclusion, considerably high levels of intrapopulation variation in heat tolerance were detected in this study, and coupled with climate action, coral populations may have sufficient reservoirs of heat tolerance to sustain their replenishment through natural or assisted evolution. Thanks for listening, and thanks to all our collaborators and funding partners.